Assalamualaikum warahmatullahi wabarakatuh. My name is Amira Nishara Iza binti Muhammad Yassin. I'm from S2446C. Now, I'm going to present my final year project titled Immunogenicity of SARS-CoV-2 Epitope Identified by Immune Epitope Database and Analysis, Iedb server. First of all, let me explain what is COVID-19. It is a novel coronavirus disease that caused by severe acute respiratory syndrome coronavirus 2. It was found in Wuhan, China in the late 2019. Hence, it was called COVID-19. This continuously growing outbreak subsequently made vaccine become urgent global demand to control the spread and gain population immunity. But the existing vaccine, which produced by the traditional method that mostly use whole pathogen, life attenuated, take a longer time and quite expensive. To overcome these drawbacks, modern vaccine development that use reverse vaccinology comes to play. Reverse vaccinology is a new approach in vaccine production which use more in computational methods and bioinformatic tools. This method capable to identify the presence of epitopes in specific sequence to develop a peptide-based vaccine against COVID-19. Epitopes play a crucial role in the immune response as they are the specific region of a virus that bind to antigen receptor on the surface of B cell or T cells. However, in this study, it will be more focusing on the binding for T cells. T cells could be divided into cytotoxic T cells by binding of MHC class 1 and helper T cells by binding of MHC class 2. And both of it will be further investigated using web-based Iedb prediction tools. Next, we move on to the problem statement. Based on research study, traditional method is time-consuming as it takes up to 10 years to develop. At once, increase the expenses. The complicated protocol cause more tedious work need to be done and introduce more to be exposed to this particular virus, which is not a safe option. In contrast, the computational method provide a faster, low cost, more accurate result and safe environment as it only need access to database tools to design target vaccine in a dry lab practice. The objective of this study are to predict the T-cell epitopes of SARS-CoV-2 protein using Immune Epitope Database and Analysis Iedb server. Second, to predict the most conserved epitopes using Conservancy Analysis App. And third, to explore the suitability of Iedb server as prediction tools in immunoinformatics study. Now, let's take a look at the methodology. First, 30 samples of spike glycoprotein sequences of SARS-CoV-2 were retrieved from the database in FASTA format. The samples are objectively chosen from local cases in Malaysia ranging from year 2020 until 2022. Second, each sample will run individually in the according T-cell epitope prediction tools. Then, the most repeated epitope sequence throughout the sample are counted and selected as top potential epitopes. These steps are aligned with the objective one. Third, those listed epitopes will undergo conservancy analysis, which also in the Iedb future tools. This helps in identification the most conserved epitopes, and it further visualized by web logo tree tools, which generate sequence logos that show results based on the similarity and conserved degree. Both steps conducted to follow the objective 2. Lastly, the predicted epitopes were compared with previous study to validate the final finding in this study at once, aligned with the objective 3. For the result, MHC1 binding tools identified 10 T-cell epitope of spike protein, and based on the peptide score, epitope 1, 2, and 3 were the most potential peptide for target vaccine as higher score indicates the binding affinity. Meanwhile, for MHC2 binding tools, 8 T-cell epitope or spike glycoprotein were identified. The potential peptide are also epitope 1, epitope 2, and epitope 3 by observation of its score. Next, we move on to the conservancy level of MHC1 binding. All epitopes substantially conserved as each one of them are over 80%, even the lowest are 93%. For further visualization in Web Logo 3 tools, epitope 1, 2, and 3 are chosen to be more focusing on as they have highest binding affinity and 100% in conservancy level. Same interpretation goes for MHC2 binding, but only epitope 2 and 3 were chosen for visualization on Web Logo as the epitope 1 does not 100% conserve. Even all epitopes substantially conserved, these two epitopes were highlighted as it may have credibility to be in target vaccine design. Then, the predicted epitope were displayed as the picture attached here in the slide. The higher the letter stack, the higher the frequency of the corresponding on that certain position. In MHC1 binding, seems that the predicted epitopes are purely conserved as no overlap or small letter observed. And in the MHC2 binding, the overall height of each letter in the specific epitope was high and it surely as it's supposed according to this conservancy result. Next, based on table 4.7 here, a list with all the overlapping predicted epitopes with the previous study are shown respectively. For MHC class 1 binding, epitope 3, epitope 9, and epitope 10 were overlapped with several studies that released from year 2020 to 2022. On the other hand, for MHC class 2 binding, epitope 2, epitope 4, epitope 5, and epitope 7 were overlapped with several studies that released from 2020 and 2021. In conclusion, by utilizing the Iedb prediction tools, the prediction of T-cell epitopes of SARS-CoV-2 could be done by analysis the binding of MHC class 1 and MHC class 2 molecules. According to the result, epitope 3 from MHC class 1 binding and epitope 2 from MHC class 2 binding are the best candidates among others which show high binding affinity with 100% conservancy level and supported with previous study which indicates it's worthy for vaccine target against COVID-19. For future recommendation, other parameters such as antigenicity analysis, comparing results from other prediction tools, and validation by wet lab practice were aspects that needs to be done more truly for more reliable outcomes. That's all from me. Thank you.